Okay, in this video what we're going to do is have a look at malted barley and the role of malted barley in the brewing process and the influence it has on the end product. So just to have a quick look over the objectives of this video is firstly to describe the key features and structures of the malt grain that is important to brewing itself. And then what we're going to do is explain how key components of the malt grain influence the colour, the flavour, the texture and the strength of the resulting beer, and how we can vary that using different malt variants and different temperatures, etc. These kind of things that we're interested in. Now, firstly, let's have a look at the barley grain itself. Now, the barley grain we've already touched on in the videos previously, but what we're going to do here is just go into a bit more depth about the barley grain structure and where we're going to get key bits and pieces from. Now, when we look up close at a grain, and we're going to cut one in half essentially here, we can see right around the outside is the bran, and the bran essentially is the husk. This is the hard substance that encases the grain. When we do the milling process, we break open that husk. In doing so, we expose the two key tissues that we're really interested in. We're not interested in any of the bottom. The germ at the bottom is not important to us at this stage. What we are interested in, firstly, is the endosperm, and second is the alurone. And these are the two tissues that we're trying to expose to the strike water that we add in the mashing process that we'll talk about a bit later on. So let's have a look at the endosperm itself. Now, the endosperm, when we look at it under the microscope, here is a scanning electron microscope image. So this is a 3D image, essentially really close up of that endosperm. We can see these kind of grain-like structures. And inside these grains, we find starch, or more scientifically known as amylose in this case. Now, the amylose is essentially a chain of glucose molecules that is coiled together into a helix. So what you can see in this image at the bottom are small little hexagons, and they're each joined by a little bond. So each of these little kind of hexagons essentially is a sugar that we want to release from this chain. We want to break that chain. And we can't break that chain in any way other than basically by using enzymes. Okay, and we'll talk about enzymes shortly. They're really important in terms of the role of breaking this chain. Now, what we want to do in order to brew essentially is break this chain into disaccharides. Now, what this means, di in science means two. Okay, so we want to break this chain into sets of two, and this is explained here. So maltose is a disaccharide. Maltose gives the name of malt. If you understand what I'm saying, malt and maltose essentially deriving from the same term, and we understand here that maltose is essentially a disaccharide in the sense that it contains two glucose molecules attached by a single bond. They look like they're holding hands in the middle. So you can see the hexagon shapes. There's two of them there. And we want to break that long chain of hexagons into two maltose molecules, essentially. So that's the thing that we need to do. So all of the sugar essentially comes from the endosperm, and it makes up about 80% of the grain structure itself. So how do we break these chains? Well, this is where we jump to the alurone. Now, the alurone here, shown at the top under the microscope, are the cells with the blue surrounding. So they're quite large cells, as you can see, compared to some of the starch molecules that you can see underneath them that are the black dots. This layer of tissue is responsible for producing and secreting enzymes, and it produces essentially two really key enzymes in this process. And the first is alpha amylase, and the second is beta amylase. Now, you can generally tell a lot about an enzyme by its name. Essentially, all enzymes end with ASE at the end, A's, and the beginning of the enzyme generally tells you the molecule that it works upon. So in this case, each molecule works on amylose. So hence we call them amylase. Now, they work in different ways. There's two for a reason, and they do tend to work hand in hand, although we can manipulate each of these enzymes depending on temperature. We'll talk about that shortly. Now, let's take alpha amylase to start with. Now, alpha amylase is a smaller enzyme of the two, and it is able to attach to the starch chain at any point it wishes to. And it kind of bimbles along. It just kind of attaches where it likes. It nibbles where it likes, and it breaks that chain where it likes. And what it tends to do is it just kind of clumsily breaks this chain into small pieces, but not as small as we want them to be in this particular case. And what it actually produces is something called dextrins. Now, on the right-hand side, you can see these kind of 4, 5, 6 molecule chains that aren't actually what we class as fermentable. Yeast isn't able to break these down. So they're not really important to us in terms of producing alcohol, but they do have other properties that we're interested in, and we'll talk about that shortly. Now, beta amylase is shown here. It's a slightly larger enzyme of the two, and it generally attaches at the ends of chains. So what it does is it will attach to the end of a starch chain, and it will break the bond between two glucose molecules, essentially, but it will leave one in the middle. And what this actually produces is the maltose that we're interested in. So what you can see on the right-hand side are lots of disaccharides, two chain molecules. And these are all maltose molecules that are really important because these are fermentable. These are the ones that the yeast will use further down the line to turn into alcohol. Now, what this actually means is that we can actually manipulate the enzymes to produce either more maltose or more dextrins. And we can see this in this particular graph here. So what this graph shows, what we're really interested in, I think on this particular graph, is the top axis is the temperature in degrees. So it runs from 50 up to about 80. So this is the temperature of the mash. And then what it shows within the graph are kind of two really important lines. Is the blue line is the action or the activity of beta amylase, and the red line is the activity of alpha amylase. Now, if we were to brew or to mash, at around 64 degrees Celsius here, we can actually produce a higher alcohol by volume. So ABV, this is the strength of the beer. So your typical kind of beer might be 4 or 5%. But if you want to increase that, then you'd need to mash at a lower temperature. This is because the beta amylase is much more active at a lower temperature. It's actually less active at a high temperature, and it produces more of those maltose molecules. So as we can see then in the green line, you can see the fermentability is higher at the lower temperature than it is at the higher temperature. Now, we don't always want to go after a high alcohol by volume. Sometimes we might want a thick, kind of more syrupy type beer, maybe something like a stout or something like that, that's quite thick, quite textured in the mouth. Then we would perhaps brew towards 70 degrees. So here, what we do is we reduce the activity of the beta amylase, and we start to increase the activity of the alpha amylase. And we actually increase the production of the dextrins, which is the pinky kind of purple line. And what this does is it increases the production of those longer chain molecules that don't get broken down by the yeast. But what they do is they give the body to the beer. So it makes it more thick, gives it what we call a mouthfeel. And you can find this again, like we said, in some stouts or darker ales that you're kind of interested in that kind of case. So changing the temperature of the mash is possible, and we can therefore impact the activity of the enzymes. Generally speaking, what we'll try to do is we'll try and balance this out. We want good alcohol by volume, and we want some body to the beer. Therefore, we'll try to brew at around maybe 66, 67 degrees Celsius. And this is what we class as the brewer's window. And what we're doing here is we're using the alpha and the beta amylase to work together. So the alpha amylase comes along, breaks the starch into kind of smaller chunks, and then the beta amylase comes along and breaks those chunks down even further into the maltose that we want. And it's important that we understand the role of both of these. So that covers essentially the grain structure. Now, what we want to understand at this point then is the influence of the grain on the beer. And the end product is influenced in the following ways. So firstly is the alcohol strength. That's a funny image. I'm going to talk you through the image in a minute. So the alcohol strength, which is what we class as ABV, alcohol by volume. Secondly, it's the texture and the mouthfeel, which we'll talk about shortly and what that means. We've kind of touched on these two already a lot. So we won't touch on these a great deal, but we will talk about them a little bit. It's the next two are really important. The next thing that the malt can influence is the colour. So here you can see a kind of range of different colours that we can get by using different combinations of malt. And again, we'll talk you through the kind of science behind that. And flavour. Flavour is really important. This is generally what we class as your base flavour. Now, in 2 sessions' time, what we will look at is hops. And hops, we understand add to the flavour and the aroma of the beer. But the base of this comes from the malt, and we'll talk about this shortly. So let's just work through each of these in turn. So alcohol strength, and this is a really controversial beer. As you can see, it's the bottle's been put into a stuffed squirrel, and you might wonder why that is. This particular beer was brewed by Brewdog. It's called The End of History, and it was brewed to be the world's strongest beer at around about 55% ABV. This is achieved in two ways. So if you were to take your average kind of IPA and you were going to make 20 litres of it, you'd need about 4.5 grams of malt to do that. Now, to make this particular beer, The End of History, you need to increase that malt to 12.5 kilograms of malt. So you're almost tripling the malt content, and therefore you're increasing the amount of malt and maltose available to the yeast. Now, secondly, the way that Brewdog achieved this particular beer is they, when they've taken the beer and they've made it, they freeze it. And the whole, it's generally frozen at about minus 70 degrees Celsius, which is really difficult to achieve. Now, as the beer thaws, it's the alcohol that melts first, and they're able to extract the alcohol and the flavours from the water. And this gives you essentially what we call an ice bock. And there's a bit of a rivalry between Brewdog and a German brewery called Schorschbrau. These guys made a 57% beer and actually took the record. And then it's been taken again by a Scottish brewery that made something called Snake Venom, which is now standing at 67.5% ABV. And again, they're achieving this by using vast amounts of malt. It'll be really expensive to make these particular beers. But we understand, therefore, that malt plays a really important role in the ABV of the beer, so the alcohol by volume. The more malt that we use generally, the higher the strength of the beer. Mouthfeel is the next one that we talked about, and this is one particular example of a beer that has a very thick mouthfeel. You can kind of see the name, the caramel fudge stout. You can kind of imagine it's going to be a very thick, cakey kind of beer. Now, how they're doing this is by really influencing the role of those alpha amylase and just making them probably mashing at a higher temperature, getting the production of the dextrins, those slightly longer chains that don't get fermented. And that gives it this kind of thickness. It's nothing to do with the colour. Now, the colour itself, here you can see a wide range of different colours that we can access by using different malts, essentially. So you've got everything from the really pale on the left to the really dark on the right, and everything in between. So you've got things like lagers and pilsners and things like that on the left-hand side. Towards the middle, you might get kind of brown ales, ruby ales, these kind of things. And then towards the right-hand side, you've got your dark stouts and things like that. Talk a little bit more about this in a minute, but essentially this colour comes from the malt. Now, the way we get the colours essentially is by changing the way that we kiln or heat the malt. So if we want a pale malt on the left-hand side, what we'll do is we'll just take the malt straight from the field. We'll malt it essentially, and then we won't kiln it. And it's not roasted in any way. If we want a really dark one, what we'll do is we'll put the malt into a kiln and we may heat it and roast it. And what that does is it causes the darkening of the malt itself. Now, this in turn influences the flavours that we can access. Now, what you can see here is on this wheel, lots of different flavours. So things like if you want a kind of sweet, mildly malty kind of base, then you'll slightly kiln the malt itself, and you'll get what's essentially at the top right hand side. If you want something that's like nutty, you'll kiln it and you'll roast it. Or if you want something that's kind of coffee, then you'll roast the barley but you won't kiln it. So there's all sorts of ways that you can kind of manipulate the malt itself, and you can look into this in more depth. But what's really important to note is that you are either taking the malt as it is, you're kilning it, or you're roasting it, or you're doing both. and this gives you different flavour combinations. And this also changes the colour. Now, beer colour is measured by what we call SRM, and this is a standard reference method, and this is shown here. And what you can see here is, I think at this point you start to realize how many different variations of beer exist, and they're all kind of determined either by colour or by strength, and all sorts of things. And you can play around with all sorts of different variations to get this. But what this gives us is a method by which we're able to give a number to the colour of the beer, so we can see where it sits. And essentially different beers have different SRM ranges. So if you're making an imperial stout is generally a very strong stout. It will have a lot of alcohol in it, but it will be very, very dark. So we're talking 50 to 80 on the SRM scale. Something like a pale ale generally will sit somewhere between 5 and 14. So that's somewhere between a kind of pale gold, maybe upwards towards a deep amber, depending on the variants that you want. And generally there's no real rules with regards to the way that you can brew. You can do whatever you want. It's generally down to your own kind of choices as to how you would go about doing this. You can play around with it and experiment. Okay, so to summarize this video, malt contains the sugars required to create the alcohol. Enzymes inside the grain convert starch into sugars. Different temperatures result in different combinations of sugar content, and in turn influence the alcohol content. Malt influences strength, or ABV, texture, colour and flavour of the end product. And varying the malt leads to different end results. So there's all sorts of ways, as you can see in this, where you can change the outcome of the beer. Once you've had a review of this, you can watch it again if you need to, make some notes, pause the video as you need to, have a go at the quiz at the end. Good luck.